In this video we will discuss about a trimeric complex that consists of ALDH4A1, MC1, MCP1 and MCP2 in together helps in the import of pyruvate in the mitochondria. So first they checked whether this trimeric complex formation occurs or not, and if we combine this size of this ALDH4 MCP1 MCP2 in the monomeric manners, then its size will come around 88 kilodaltons. So they checked first for this MCP2, MCP1 and ALDH4A1. All three of them are extracted, or Western blot of all immunoprecipitant. Western blot of all three of them shows a similar size, indicating that there is a trimeric complex formations occurs. Then they did the chromatography, and here also they see all the three agents are come out at the same time, indicating that they are forming a complex, come out at the same fractions, that we can see that. So indicating that they are forming a trimeric complex. And thereafter, using the alpha fold 2, they wanted to check which regions of this ALDH4A1 helps in the trimeric complex formations with MCP1 and MCP2, and they are able to find out that around base pairs like amino acids from 191 to 198. This is the approximate region. You can check the exact range in the paper, and this range helps in the direct interactions with this MCP1 and MCP2 that are shown here in the panel C, and these analyses are done on the alpha fold 2. So thereafter what it did is, and these results are also in line with the previous results, that shows that if this region is mutated or removed, then it will reduce the pyruvate import ability of the cells. So now what it did is, they check the complex formations. They check the complex formations by immunoprecipitating this ALDH4A1 wild type, and so here the Western blot we can see that ALDH4A1 wild type signals are there, and thereafter if they use the reintroduce this mutated wild type, they see very faint signals, and when they check this MCP1 and MCP2, they can find their signals are also very faint. In the case where this mutated ALDH 4A1 is used, and now they developed this proteoliposomal proteoliposomals method to check the effect of this MCP1 MCP2, or when both are present, and when both are present with wild type ALDH4A1, or when both are present with wild type mutated ALDH4A1. So this is the representations of proteoliposomals method, and here the Western blot shows the different conditions that they have used for the analysis to check its pyruvate import, its effect on pyruvate import. And what they're able to find out is that when this either MCP1 and MCP2 are present, then there will be increase in the pyruvate import, but those increase are significantly enough compared to the case, yeah, and those imports are significantly up compared to the control case. And when they used both of them, the MCP1 and MCP2, the increased in the pyruvate import increased slightly more. But when they use MCP1 and MCP2 with the wild type ALDH4A1, the increase is maximum. And when they used mutated ALDH4A1 with MCP1 and MCP2, they also see an increase similar to the wild type reintroductions of ALDH4A1, indicating that this ALDH4A1 helps in this trimeric complex formations that in return upregulates this pyruvate uptake. And when they check the kinetics to check the half-life time period. They are able to see that when the wild type ALDH4A1 is used along with MCP1 and MCP2, or mutated ALDH4A1 is used along with MCP1 and MCP2, they see a least T half time period compared to the empty vector case or case where they have used MCP1 and MCP2. So that's all for this video. I hope you understood that how they come up to a point that this trimeric complex formations occurs, and that helps in increasing the pyruvate import. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.